All right, so let's continue on. So the next organelle is lysosomes. Have you heard of those before? What comes to mind when you hear that? What do they do? Yeah, they digest, right? Scientific terms, garbage disposals of the cell, whatever you want to say, right? So lysosomes are important for quite a few different things that we'll talk about. I'll come back to this graphic in a second. Let's look at some of the verbiage here. So they can get rid of damaged, foreign, or unneeded molecules in organelles. Now, they are involved in intracellular elimination, but they can also help the cell after it's engulfed something. We'll talk about macrophage for a second. But one thing to keep in mind, the lysosomes kind of create a microenvironment that's really harsh. So they're enclosed structures. They have lipid bilayers that on the inside of the lysosome, which is known as the lumen, right? The lumen is the inside or opening in something. So that's their interior. It's got a pH of about 4.8. What's that relative to a cell? What's typical cellular pH? About 7. All right, our body overall has a pH of approximately 7. Our blood pH, remember what that pH is? 3. Yeah. 2. Yeah, 7.35 to 7.45 is blood pH. It's extremely important that it stays within that range. So it's regulated by a few different pathways or processes, I guess you'd say, mechanisms. Why is it so important that our pH stays about that? It's one of the main reasons. What depends on that pH? Yeah, protein structure. Right? As soon as you change that pH, you change the thermodynamics of protein structure. In other words, some of those amino acid interactions are pH dependent. As soon as pH changes, those proteins can denature. So if you hear these things saying, hey, eat this diet, it changes your body's pH, go under this particular type of therapy because cancer can't grow at this pH, whereas your cell can, you don't change your body's pH. Okay, so it's something that we have to be careful of how that is stated and exactly what they're driving at. Our body overall stays in that neutral pH, and the lysosomes do that on purpose so that when proteins go in, they denature. So they use a combination of a low pH and digestive enzymes in order to degrade these molecules. It contains about 36 digestive enzymes to break down proteins, polysaccharides, nucleic acids, and phospholipids. Right? So some molecules go in there on their own. Some molecules end up as part of organelles that were surrounded by lysosomes. So there's a lot of different molecules and structures that are degraded by lysosomes. And here's some of the things that happen. Specific catabolic lysosomal processes. They can remove cellular proteins if it's damaged or no longer needed. Our proteins tend to stay around for a relatively short period of time, and as they function inside the cell, they can go through changes that would warrant their removal. If they're no longer effective or we don't need them anymore, they're removed. And one of the reasons our body does this is so that we can adapt. If we made proteins in response to certain nutritional or environmental stimuli, and those are no longer present, we need to be able to remove those proteins that we no longer need. So that's what lysosomes are important for. That's part of it. They also are involved in phagocytosis, right? Phage means to consume. So this is essentially cell consumption. Some cells are able to engulf other cells, and then they break down the foreign substances taken in from outside themselves, and this is used by the immune system. And which cells are famous for this? Macrophage. Right? Macrophage are large cells. They engulf other molecules in cells, and they're loaded with lysosomes. And then they degrade those molecules before it causes systemic harm to us or other animals that use this system. So they do that. They're also involved in autophagy. So autophagy is the elimination of organelles. This is from within the cell that have been targeted for breakdown. For example, mitochondrial turnover rate in the liver is about every 10 days. Mitochondria are made, they function for about 10 days in the liver, and then they're replaced. And one of the things to keep in mind is all those electrons that are moving around as part of ETC, it's something like 0.1% of them don't end up where they're supposed to, and they damage other molecules. And so the mitochondria sustain damage from their processes over a period of time, and so they're just eliminated. And so the lysosome would engulf the mitochondria and basically digest them, and so new ones can replace it. The last one here is bone demineralization. Bone cells use lysosomes to degrade bone to release stored minerals into the bloodstream. And the classic example is for calcium, right? So what are the bone cell types that do that, to degrade bone? Osteoclasts, yeah. They have lysosomes, and osteoclasts use a combination of acids to mineralize the bone, and then they use lysosomes to degrade part of that. And then they'll put calcium into the blood from the bone. And calcium is so important for things other than skeletal structure that our body will break down our bone in order to keep calcium maintained in the blood. It's important. Calcium is even crucial for things like insulin release. If blood calcium is consistently low, it can affect insulin sensitivity, for example. There's lots of things like that. So it's very important that a person consistently consumes adequate calcium in the diet, or you're breaking down bone in order to maintain blood calcium. So sometimes the best way to assess a person's long-term calcium status is measuring their bone density. I think this has things we talked about. Phagocytosis. This is a cell that contains lysosomes. Autophagy is a damaged organelle, like a mitochondria, is infusing with the lysosomes. So the lysosomes can kind of, these structures, their plasma membranes can kind of be pushed into the lysosome, and their plasma membranes will kind of merge, and then their contents go into the lysosome. So lysosomes also are consistently being turned over. And we'll talk about this one later on in the quarter, in a few weeks, probably 3 or 4 weeks. Receptor-mediated endocytosis. Have you heard that before? That's a mouthful, right? That's where receptors on the surface of the cell basically grab molecules, and then the cell engulfs them, and then the lysosome separates out those molecules so the cell can get what it needs, but eliminates things of that process that it doesn't need. We'll talk about LDL receptors when we talk about that, and blood cholesterol. Good there? Nucleus. If this is your favorite organelle over and above the mitochondria, that's a pretty good argument, right? So we're going to talk about this in the context of how nutrients actually affect nuclear events. All right, so a little bit about the overall structure. The nucleus has 2 membranes also, and so they call it the nuclear envelope. And then what happens with the nucleus is you have proteins that form these kind of pores, these nuclear pores, so that molecules can go in and out. But the passage of molecules through those pores is policed. It's monitored by these proteins. So they help some molecules go through, either going out or in, and they also stop some other molecules going through it. What else? The chromatin, right? Chromatin is a combination of DNA and protein. And the nucleolus, remember what that's important for? You heard that? That's ribosome synthesis, nucleolus. That's where ribosomal RNA is made, and then ribosomes are made. That's why it's a dense portion, the nucleolus. What else? All right, here's the endoplasmic reticulum. It's got ribosomes, so this is really RAR. Right? It's closely associated with nucleus, and we'll see why probably next class. Is because when mRNA is leaving the nucleus, some of it's translated, and as it's translated, the proteins are put on the inside of the ER. So it's very common for proteins to their mRNA to come from the nucleus and go right into protein synthesis that involves the ER and the RER. So we'll look at that pathway. But again, nuclei are pretty distinct structures. You can easily purify them from cells, just like you can from the mitochondria. The ER and some of the other structures are not as easy. Lysosomes are not as easy, for example, to isolate. All right, some verbiage. Largest of the cellular organelles. The nuclear envelope is double membrane with pores. Last passage molecules such as mRNA, DNA replication. So some terms, right? DNA replication is the copying of DNA. Remember, that occurs before cell division. And transcription, we talked about that. That's synthesizing RNA from DNA. And then translation is RNA to protein. You're translating from the nucleotide language to the amino acids. The presence of genes allows the nucleus to initiate and regulate most cellular activities. We'll look at that. And the DNA in all cells is identical. However, different cell types arise from differential gene expression that yields different proteins that allow cell types to be specialized. For example, liver cells may need to make enzymes or proteins that skin cells do not. Those two cell types have the same nucleic acids, right? They have the same genetic material, but they express their genes differently, and that's what makes the cell types different. And the classic, you guys probably weren't around yet when Dolly the sheep was cloned, and the main kind of thing to come out of that for the general population is what? Oh, we can clone an animal. Scientists said, no, that's not why we did it. Why'd they do that? To prove that all cells contain the same DNA, essentially. To prove that somatic cells, it didn't have to be gamete cells or cells that are normally involved in reproduction. So in other words, they took epithelial cells from a sheep, they isolated the DNA, and then they took an egg cell and they removed its genetic material, put that somatic cell DNA in there, and then fertilized the egg and got a complete animal out of it, right? That's cloning. And and I remember when that paper came out in Science magazine, and then I saw it in the media, and I went, wow, they're talking about the wrong stuff. What that means is, you know, like a liver cell and a muscle cell make different proteins even though they have the same DNA. So what we want to learn is, how do those different proteins affect their metabolism or their nutrient requirements? So as we go through this, keep in mind, like for example, when I say, you know, beta cells have GLUT2, muscle cells have GLUT4, they're expressing different genes. Even though they have all the same genes, they differentially express them. So that affects a lot of things related to nutrition. But the numbers are staggering, right? You guys were also around, remember when they finished the Human Genome Project? What was the point of that? What did that project do? You guys are familiar with the Me-23. What's that, right? 23 different chromosomes paired, so we have 46 chromosomes. Me-23 means it's kind of a genetic study, right? And they'll do things that they think will interest people, and they'll give you information on those genes. The Human Genome Project sequenced the entirety of the human genome, all of the nucleotides. So I was in graduate school when they started this, and I was familiar with, originally it was going to be, hey, our lab against your lab, against this lab, against this lab. And then they got into it, and they went, there's a lot of work, can we cooperate? And then labs around the world started sharing DNA sequences, and what it did for us was, it meant that I could now go from, when I was first in graduate school, in order to sequence something, we'd sequence ourselves. We'd pour these big long DNA gels, and this is all history for you guys, and we'd read the DNA sequence, and we'd go through all this thing, and we'd piece together these genes. And then later on, when I was at Portland, like in a research position before here, I used to take my DNA that I need sequenced, I'd walk across campus and hand it to them, and 2 days later I'd get an email and go, there's your sequence. It's phenomenal how different it is now. A lot of that technology came from this. One of my other questions was, well, whose DNA is it? It's not mine, right? Everybody's DNA is slightly different. That's why we have different nutrient requirements. That's why we look differently, we behave slightly differently. Our genes are slightly different. So as we go through this, I'm going to ask you things like, tell me how a gene sequence, tell me how the sequence of a gene that is different between person A and person B affects this process. So we're going to talk a little bit about genetics in here and how it affects our health and nutrient requirements. Well, one thing to keep in mind is we're talking about billions of nucleotides. It's probably 4 billion nucleotides in the human genome, all in a sequence that determines all of those different things. Only a small amount of these bases are part of genes. Most of it is what they used to call junk DNA. It's the sequence in between genes. And now a few years ago, they started realizing, let's not call it junk anymore. It's extremely important for regulation of gene expression. But one of the questions is, if you've got all these nucleotides inside a nucleus wrapped around proteins, how are the genes identified? And then how are they expressed? So we're going to review some of the things that you guys have talked about in other classes. Things like that it's actually proteins called transcription factors and enzymes that find specific DNA sequences. And they bind to them. And sometimes it's a chain of events that now causes a gene to be expressed. What's interesting for us is some of that process regulated by what we eat. So the regions that are part of this process that are called promoters. So for us, think of a promoter as a noun. So a promoter is a nucleotide sequence that regulates gene expression. So make sure you put that in your notes. A promoter is a nucleotide sequence that regulates a gene's expression. Most of the time they're in front of a gene, right before the transcription start site. And once proteins start binding to that promoter sequence, you can accumulate a transcription complex. So we'll talk about that. So I think we're going to go through some more review. Oh yeah, I don't know why I did this like this. DNA to RNA, transcription, you have translation. The entirety is known as gene expression. And in general, the more RNA you have, the more protein you're going to have. We'll talk about some different levels of regulation. Right? DNA is unique. So that means we can have possible differences between people. Basic differences between DNA and RNAs. Right? And you guys have all read the textbook. You know that it talks about these details. So I expect you to remember these. So I'll ask you a few differences between DNA and RNA. And one is just the fact that this group, this hydroxyl group in DNA, has lost an oxygen, so it's deoxy. Here's RNA. It's got an OH group. What's one thing that does? Yeah, it destabilizes it because this oxygen can perform nucleophilic attacks on adjacent nucleotides. So DNA is a lot more stable than RNA. We know that. We've all seen Jurassic Park, right? So I won't tell you a story about that. That's the pathology. Okay? So that's one thing. The other thing is DNA double-stranded. In general, we always say DNA functions as double strand. That's another thing that improves its stability. Right? It's a double strand wrapped around proteins. What's the name of those proteins? Histones. And we're going to skip that level of regulation, that whole condensation, decondensation. Right? We're going to assume that the genes are accessible. What else? Now RNA is single-stranded, but so a lot of times when we draw RNA, especially me, I'll do this too. So, oh, that's mRNA. But really, that RNA can have all types of different structure. Right, but it is single-stranded, but sometimes it folds on itself and does stuff like that. You'll talk more about that in 329, I hope. That's one thing. Another thing, nucleotide bases are different. DNA has thymine. RNA uses uracil instead. That's another characteristic. What else? Yeah, so RNA can be degraded very easily. If you start working in an RNA lab, you have to be careful because any contamination of anything in your RNA degrades. When we first started working on it, we did the full overkill, and then later on we realized, yeah, just make sure your water's clean. So, but it's a little bit different. I used to have the students in 329 lab do RNA isolations, and almost every time they worked. And then we do reverse transcription PCR. And we'd look at expression of certain genes based on certain nutrient levels. I don't do that anymore because I think I enjoyed it more than all the students. So I went, all right, I'll get over it. But it is a completely different molecule, so make sure you remember those differences, right? And then the basics of the central dogma of DNA, right? There's the DNA double strand. It's got direction, which just means basically that this is kind of your open end. All right, it goes 5' to 3'. In this case, your 3' end has that open hydroxyl that another nucleotide can bind to, right? And DNA forms that complementary base pairing. So A and T combine, GC combine. How are they linked together? What kind of bonds? Hydrogen bonds is what stabilizes that. And if we look at it, it's transcribed, right? So we have a sense strand of DNA and an antisense. The sense strand is essentially the one that is in the mRNA. Makes sense, right? By copying, making a complementary copy of the antisense, you're copying the sense strand sequence. The difference, you're replacing the T with a U. So you can see a UG, right? Now, once you have an mRNA molecule, we'll talk about modifications in a second. Now we can do translation. And the translation is based on that nucleotide sequence of mRNA in sequences of 3. Each one of those sequences of 3 is called a codon. And so if we look at the genetic code here, there's a couple interesting points. And one is, right, we know methionine is always the first amino acid, right? It's got the initiating code, it's an initiating codon. That first tRNA is the one that will start this process. And so it always starts with the methionine. But if we look at this sequence here, one of the interesting things is the redundancy of the genetic code and how it affects us. So if we look at this sequence, let's look at down here at the bottom. So if we say, here's our first base, second base, third base, and we say, for example, let's say one of the enzymes that I have, its nucleic acid sequence at a specific location of that protein, right? Let's say, yeah, here, okay, is GCG for me. So my protein in that enzyme, it's an alanine. And remember the alanine side chain? It's a methyl group. Doesn't do a lot. Small, not steric hindrance, not charged or anything. Okay. Okay, so that's me. Let's say in most other people, rather than a C, it's an A. Okay? That means that in most people's the enzyme, rather than an alanine, that's a glutamate. Remember the side chain of glutamate? Also known as glutamic acid. It's a carboxylic acid group. It's charged. So now I go from an amino acid that doesn't do a whole lot to a charged one. That can change the structure of the protein. So a single nucleotide change can cause a significant difference in the protein. I go back to my example I talked about before, that mitochondrial protein that we found exports iron from the mitochondria. One nucleotide change caused that protein to become completely dysfunctional. because that was the enzyme. It actually was it was a glutamic acid that was required to hydrolyze ATP. So individuals that had that alteration, that protein was dysfunctional. One nucleotide change. Now, a lot of times this is what happens. Let's say mine's GCG, but everybody else is GCA. Doesn't matter. There's redundancy in that genetic code so that a difference in nucleotide doesn't always cause a difference in the protein. Okay? So you can look at it that way. Another way to look at it would be if you have a single nucleotide change, it might change the amino acid, but if the characteristics of the amino acid are similar, a lot of times it doesn't matter. We're going to start talking about nutrient-regulated gene expression and how some genetics may play into all that. And you're going to tell me things like, this could have a significant impact, or this could not because of these reasons. Okay? Because the funny part of all this is, now what's expected is, okay, nutritionists, tell us how much, a specific amount of this specific nutrient that everybody needs. Good luck, right? We hedge our bets in certain ways using population statistics and things like that, realizing that there's people that are outliers. So the RDA, which stands for RDA stands for, this is recommended dietary or daily? Got about half. Dietary. Recommended dietary allowance. What's that based on? The EAR. The EAR is estimated average requirement. Remember all these DRIs? We're in DRI land. Dietary reference intakes. So read that handout that I posted on PolyLearn. It's got a little sheet that talks about this. Okay? So one of the points of the RDA is this is the amount that will cover the needs for what percentage of the population? 98%, 97.5, not 100%. So we determine about what is the amount that's approximately adequate for half the population. Then we add 2 standard deviations. That gives us the RDA, and we're still not at exactly an amount, right? But the RDA, for the most part, is a pretty good guess on what's going to be an adequate amount for people, okay? But you can see that genetics might actually have a play in that. In order to do that, though, we have to have a way of determining whether or not you're getting enough of that. So we need a specific assay to say, yes, that's the right amount of vitamin K for you. And if we're not exactly sure how to determine that, we go, let's call it an adequate intake. And then you say, okay, the adequate intake for vitamin K is this. Somebody says, why isn't it an RDA? Yeah, because we're not really sure. So you see in the DRIs that they incorporate some leniency, but it's for some of the nutrients, it's hard to tell a lot of people exactly how much they should have. Vitamin D is a classic example. We're still doing this. Oh, do the blood test. Well, what should the blood value be? It's kind of weird. And I was in vitamin D research for a long time, so that's why I'm like, yeah, I know exactly. We're doing this. They go, oh, you can do blood tests for it. They'll say, oh, I take vitamin D. How much should I take? I don't know. What's your blood work say? Oh, I have blood work done. Who told you to do the vitamin D? Oh, this healthcare professional. Oh, why'd they tell you that? Oh. Do you know vitamin D can be very toxic? No, I didn't know that. Do you know where your level is in the blood right now? No. How long have you been taking that significant amount? I don't know, a couple of years. Right, so we want to know, kind of get more specifics on what people need, and a lot of that's based on the DRIs and specific assays that tell us how much nutrient a person should have. So we want to be careful about that. That's why in here I'll be consistently drilling down to you. And then what do you say? And then what should you do? And then what do you tell them? Because we need to be different than most people that say they're nutritionists. Right? We got to base our stuff on evidence. So that's why we go into details like this. Gene expression, all the fun stuff. Maybe I should edit all that out from YouTube. All right. So let's talk about gene expression in a little bit more detail. Remember this, a minor groove, major groove, all that, the DNA stuff, where you're like, this is biochemistry or 161? Wherever you went through any of that stuff? And you just went, it's fascinating, right? So here's a protein. And this protein is using its amino acids to bind specific nucleotides, right? So in other words, this protein knows the nucleotides it's looking for. So protein DNA interactions depend on bonds between specific amino acids and specific nucleotide bases. And the interactions that allow those amino acids to bind to those nucleotides are what kind of bonds? Hydrogen bonds are the ones that allow this to happen. So look at it this way. A change in the protein shape can alter the protein DNA interaction. In other words, this amino acid, you can see what this protein does. See where the color changes slightly? That's a different amino acid. This amino acid right here, its side chain is able to bind to a specific nucleotide here. If this nucleotide is different, let's say between me and you, changes this interaction. If this amino acid is different, changes the interaction. So when we think about genetics, we're not only thinking about the nucleotide sequence in the actual gene, we're thinking about the amino acid sequence of the proteins that interact with that gene. So there's lots of layers to this, and we'll kind of expose those as we go through it. Now, here's another kind of thing to think about. This protein structure, right, you can see these, that represents a what? Alpha helix. There's only certain amino acids that will allow a protein to form that alpha helix, right? There's certain amino acids that will do that. You throw a proline in there, breaks down the alpha helix, okay? So you've got this amino acids, you got this structure here. These are usually beta sheets, right, that are running in that way. Here's the point. If you change an amino acid, it might be way up over here, could still change protein structure. And that affects this. It's amazing any genes work. So all these different things that it relies on. Okay? And these are interacting within the major groove. Here's the minor groove, major groove. So there's only certain parts of DNA that they'll actually bind. And remember, we're not showing histones that have to do with the wrapping and unwrapping of this DNA either. That's a different level. That's fascinating. We don't have time to go into that. Okay? So here's the other interesting thing. Some of these proteins bind nutrients before they bind genes. Some proteins rely on nutrients to change their structure or their shape, and that allows them to bind to DNA. So in other words, nutrient status affects gene expression. And we're going to look at that coming up. Have you seen a picture like this before? This is like the map of a gene. And I've got a picture here that shows this, and we'll go through some of the details. I'll kind of let me just define some things today, and then next time we'll go through the sequence of events of how this happens. So in context, let me say this, make sure everybody hears it. I expect you to be able to draw a diagram and explain the process of gene expression. So I would get in the habit of, in your notes, like drawing this and redrawing it and explain it. And anytime you have a diagram, if you want to abbreviate something, that's fine, but you have to define the abbreviation at least once. So for example, and let's not do this yet, but this is VDR. On your diagram, you can put VDR, but off to the side, you've got to go vitamin D receptor. RXR, retinoid X receptor, right? So you got to describe it at least once. Okay, so that's expectation. What else? So this diagram is similar to this. That looks like a mess, though, doesn't it? So the other one's more linear. This one has the proteins bound, and this one does not. Okay? But in your notes, let's go through some of this background here. So you can imagine this is what we're talking about. You've got this long sequence of DNA. right? Let's say this DNA is wrapped around a histones. Let's say this is 1,000,000 nucleotides long. Very easily the sequence that we're talking about. Now, out of all of this sequence, proteins have to be able to find the genes. So of all this sequence, there's a gene. That might be a few thousand nucleotides long. And then here's a gene. Proteins are consistently looking for these genes, and one of the things that they're looking for are transcription start sites. So proteins are scanning along DNA, and they're looking for areas like this. So they're looking for DNA sequences that they recognize. And those sequences that they're looking for are called elements. Remember that term? So in your notes put an element is a short nucleotide sequence. If they are in a promoter region, they can either be called promoter elements or response elements. So you can say promoter elements or response elements are those found in promoter regions. And then you can say proteins known as transcription factors bind specific elements. Okay? For example, this is where protein binds. All of these little rectangles, that's where proteins are binding. Those are response elements. So there's going to be a different protein bound to each one of those. And that's what they're looking for. This right here is the -30. It's yellow because that's the Tau box. Remember that? What binds in the Tau box? Tau binding protein. The Tau box is called that because it's like a TA repeating sequence. And it's like 9 to 15 nucleotides. They're all A's and T's. And it's a relatively consistent sequence. Once Tau binding protein finds that, it binds. And a lot of times, that's the first protein at the party. And it recruits other proteins to this. So we'll go through that sequence next time. What's +1? +1 indicates the transcription start site. That's the first nucleotide. Okay? So this is a gene that shows important sequences for transcription. Right? So at some point, RNA polymerase is going to bind. And then RNA polymerase is going to transcribe through this gene. And it's going to make an RNA molecule. Right? So what is the difference between exon and introns? Exons determine amino acid sequence. Introns do not. All right, so exon, part of protein expression, introns, intervening sequences. Remember that? Right, so when RNA polymerase goes through, and again, there's a little video on the PolyLearn, a link to it that shows transcription. And you'll see these proteins binding to the near the transcription start site in the promoter region, and then you'll see them be activated, and then RNA polymerase starts running through that gene, and you'll see all the nucleotides coming into RNA polymerase and making that mRNA molecule. RNA polymerase will run through this gene until it hits its transcription stop site. It stops, eventually releases, and then this can be done again. Sometimes it's right after each other. So anyway, we'll look at all that. Okay? So next time we'll talk about general transcription, and then we'll talk about why they put vitamin D in milk. And that's what this has to do with vitamin D gene expression. Okay, I'm good. Any questions about that? Okay, guys. So what I'll do is I've got to convert these, and once I start converting these videos and I put them up, I can start sending you guys the links in case you want to go back through them. Okay, guys. So I'll see you over in the other classroom on Wednesday, right? Building 3, 104.
Could you keep reading some of this up? Like pages that we need to read?
1 second.